In this video lecture, we're going to look at blood typing. This is the last of the videos on blood. And so we'll start out by comparing agglutinogens and agglutinins and how they determine blood type. And then we'll look at two blood type groups, the ABO blood group and the RH blood group. And then last, we'll look and see what happens when you mismatch blood. And then, of course, how do you determine a person's blood type? So let's first start with the blood types. On the red blood cells, what are called antigens. These are also called agglutinogens. They are basically markers on the red blood cells to indicate that this blood belongs to you. It's a glycoprotein that's on the cell membrane. So I want you to think of it as dog tags. Just little markers, again, that just say, hey, this is your cell, not somebody else's cell. Plasma antibodies are also called agglutinins. These are the gamma globulins we've talked about before, and they recognize foreign antigens, and then they deal with those by sticking to them and causing them to bind and therefore agglutinate or clump. So that if you have a mismatch of blood transfusions, say you're A blood and you get a transfusion of B blood, your antibodies will attack that B blood and cause it to agglutinate. There's a lot of different blood groups out there. There's 30 blood groups. We're only going to look at the ABO and RH blood groups because those are the ones that cause the most severe transfusion reactions and therefore have a clinically significant problem if there is a mismatch in blood. Other blood groups don't typically have as severe a blood transfusion. They're only going to see any kind of transfusion reaction if you have to have a huge amount of transfusions. So let's start with the ABO group. Now imagine I have A positive blood. So when I refer to the ABO blood group, I'm talking about the A part. The plus or negative of someone's blood type refers to the RH group, and that's going to be dealt with separately. So your ABO blood type is going to be determined by the presence or absence of the agglutinogens or antigens that are on the red blood cells. So here's the four groups or four types of blood. You can either be A blood, B blood, AB, or O blood. If you have A blood, then you have A antigens or agglutinogens on your red blood cells, and that's what these little glycoprotein lollipop things look like. So notice that the person has A antigens. A B blood, or a person with B blood, has B antigens on it, so you can see the plus with the antigen B. A person with AB blood has A and B antigens on every single red blood cell. So notice they have both A and B antigens. A person with O blood has no antigens on their red blood cells. So notice the negative corresponding to the absence of A and B antigens. Now, agglutinins are the antibodies. These are the gamma globulins that will attack the antigen, and they're named according to what antigen they attack. So B antibodies or anti-B would attack B antigens, anti-A or A antibodies would attack A antigens. So if you've got A blood, you have B antibodies. Obviously you don't want to have A antibodies, then your blood would attack your own blood. That would be bad. So A blood would have B antibodies. B blood then has A antibodies. You can see here, and again corresponding down at the bottom. AB blood, since they have both antigens, you can't have any antibodies. Either having one or the other would mean those antibodies would attack your own blood, and obviously you can't have that. So notice there's no antibodies in AB blood. O blood has both antibodies. Since O blood doesn't have any antigens, they can have both antibodies, and therefore either antibody will attack A antibodies will attack the A antigens, B antibodies will attack the B antigens. Okay. The agglutinins appear at about 2 to 8 months of age, and then you have those for the rest of your life. Now you don't have agglutinins that would react against your own agglutinogens. As I mentioned, you don't have A antibodies if you have A blood, because then you attack yourself. Now, what these antibodies do is shown in this picture. Here's an antibody here. The antibody has little arms on it, let's say. We'll call them that. We'll see what the antibody structure looks like when we do immune system. But those little arms here, they can attach to the antigens that are found in the red blood cell. So if this was an A antibody, that little arm would attach to the A antigen. And here's another arm that would attach to another A antigen on that red blood cell. So since these antibodies have multiple arms, they can attach to more than one red blood cell. And so when you get a group of those antibodies attaching to multiple blood cells, you end up with this blood agglutinating or clumping. Now, this is different than coagulation. When we looked at coagulation, that has to do with blood clotting. That's fibrin network. Now, nothing going on here with fibrin or platelets. This is called agglutination or clumping, where it's just the red blood cells are clumping. Now, what's the problem with this? Well, agglutinated red blood cells could end up blocking blood vessels and rupture, and then the free hemoglobin can interfere with the kidney function and could cause kidney failure. So obviously that would be really bad. So we don't want to mismatch blood. So here's a picture showing this process. You've got here a person with type B blood, which means they have A antibodies. Let's say this person got an injection of A blood or donated A blood. Now that A blood then has A antigens on it, and so the B blood's A antibodies will attack that donated blood and cause the agglutination. And you can see here it's blocking a small vessel, and that's where we get into problems. Now, the concept of universal donor and recipient is rather obsolete these days, but I like to point to why they could work in a emergency kind of situation. AB blood has been called the universal recipient since it doesn't have any antibodies. Remember, AB blood has A and B antigens, and so it can't have any antibodies. So if you have AB blood, you could get a transfusion of A blood, and then since you don't have any antibodies, your blood can't attack the donated AB blood, or excuse me, the donated A blood. O blood has been called the universal donor since it lacks antigens. Again, let's say you have A blood and you get a donation of O blood. Well, having A blood, you have B antibodies. But O blood doesn't have any antigens, so there's nothing for your B antibodies to attack. So anybody can get O blood. The problem is, though, that the donor's blood has antibodies. So if I get O blood donated to me and I have A blood, my B antibodies may not be able to attack the O's blood because the O blood doesn't have any antigens, but the O blood has A and B antibodies. And so the A antibodies from the donated blood could attack my own antigens, my A antigens, and that would cause agglutination, could be some problems. Now, typically, you only get a little bit of blood donated to you. You're not going to get a whole blood transfusion. You're just going to get maybe a pint. So the amount of blood donated to you and the antibodies that would be in the blood donated to you isn't at high enough concentrations to really cause any problems. But there's no point in doing that either, since we can match blood pretty well nowadays. Also, a solution is that typically donated blood can be packed cells with minimum plasma. If I remove the plasma, then I'm also removing those antibodies, and so there's less chance of any antibodies in that donated blood interfering or agglutinating the person's or the patient's blood. Now let's look at RH group. Now, RH group is whether you're positive or negative. RH agglutinogens were discovered in the rhesus monkey, so that's why they come up with RH as the name of this group. There's all kinds of different RH groups. The most common one is the DRHD group, because that was the one that was first discovered, and so that's the one we're going to pay attention to. In this case, a person that that's Rh positive means they have Rh antigens on the red blood cells. So a red blood cell is going to, depending on if it's AB, or O, it's going to have those antigens. And in addition to those antigens, it will have, if the person's positive, they also have Rh antigens. A person who's negative doesn't have any Rh antigens on it. Again, if they have A, B, or O blood, they may have the A, B, or O antigen, or excuse me, the A or B antigens, but they also don't have, then if they're negative, they don't have the Rh antigens. Now, if a person's Rh negative, they're only going to make agglutinins when they're exposed to Rh positive blood. So a person who's Rh positive, obviously they have Rh antigens, they're not going to make Rh antibodies against it because that would fight their own blood. If a person's Rh negative, then they're going to make Rh antibodies, but again, they're only going to make it if they're exposed to Rh positive blood. And that's different than the ABO group. Remember, the ABO group, you're pretty much born with the antibodies in place. This one, you could go your entire life if you're negative, and if you never get exposed to Rh positive blood, you'll never have the Rh antibodies in your blood. Now, the frequency of the Rh antigens varies with ethnic groups, but in the United States, on average, about 85% of Americans are Rh positive, so being Rh negative is rather rare. And that comes to another disorder called erythroblastosis fetalis, or it's also called hemolytic disease of the newborn. And this has to do with women who are Rh negative if they carry an Rh positive fetus, or if they get a blood transfusion that's Rh positive blood. Now, this describes what's going on this slide, but I'm going to flip to the next slide to show a picture of it, and then you can refer back to this slide to get some of the wording down for it. So again, this happens when the mother is negative. Now, the father has to be positive. That's genetically the only way you can end up with a positive child. You've got to have some source of that positive gene. But we're not going to worry about the genetics of this. We just want to see what happens when a mother who's negative carries a positive baby. And this is a scenario that happened in my family. So I'm going to use the Johnsons as an example. My maiden name is Johnson. My mother was Rh negative. My dad is Rh positive. And then I've got two older brothers. Dan is the oldest, so he was Rh positive. Eric, Rh negative. And then me, who's Rh positive. Okay, so that's kind of the scenario. So let's walk through what happened in my family, or could have happened, really, in my family. So here's my mom pregnant with Dan, the oldest kid, her first child. So mom's negative, Dan's positive. Now, when the baby's born and the placenta pulls away from the wall of the uterus, often you get some blood mixing between the mother and the baby. So that means some of Dan's Rh positive antigens got into mom. That means she makes antibodies, and that's what the diamonds represent here, are the Rh antibodies. So now she's been sensitized, they call it. She's been exposed to Rh positive blood, so now she's got Rh antibodies, so she's been sensitized to Rh positive blood. So Dan comes out fine because the fact that he was born before she had the antibodies made, he's not going to be affected by mom's antibodies. So he comes out okay, which of course is left up for family debate, but let's just say as far as Rh factor goes, he's okay. Now, mom's got antibodies, okay? Now, let's say we go to Eric, the second kid. Now, Eric is Rh negative. Now, he's not in the scenario up here, but I just want to mention, since he's Rh negative, it doesn't matter that my mom has Rh antibodies, because there's nothing to attack on Eric's blood because he doesn't have Rh antigens, so there's nothing for those antibodies to attack. So he comes out okay, and that's actually even more debate in my family, but you get the idea. He, as far as Rh factors go, he's fine because he doesn't have antigens, Rh antigens, on his red blood cells in order for my mom's antibodies to attack him. So he comes out okay. Now it's me. It's my turn. I'm Rh positive, so I'm over here. Mom's got antibodies made. Her antibodies will cross the placenta and they can get into me. And her antibodies then would attack my Rh antigens. And then I get into trouble. So you can actually have the fetus having severe anemia, toxic brain syndrome, and the toxic brain syndrome is from excessive bilirubin in the blood. Or it can happen during fetal development, but it can also happen at birth again when that placenta breaks away. Some of the mother's blood will get into the fetus and result in some problems. Typically, treatment, if this happens, is phototherapy, just like any kid that comes out jaundiced. They put them under the lights, and that helps degrade the bilirubin so you don't get the toxic brain syndrome going. And you can give them a transfusion to completely replace the infant's blood and kind of flush those antibodies from the mother out of the baby. And so the baby can come out okay, but if you're not aware of the situation and know what's going on, that could be very severe for the fetus, and in some cases it results in miscarriages for that second Rh positive kid. Well, now to prevent this whole thing, we're going to give the mother RhoGAM, and that's mentioned at the bottom of the previous slide. RhoGAM is basically Rh antibodies. So back here at the first kid, when my mom's pregnant with Dan and mom gives birth, those Rh antigens are now in my mother. Before my mom had a chance to build up antibodies, if she's given a shot of RhoGAM, the RhoGAM will bind up those Rh antigens and get rid of them before mom has a chance to even make antibodies. In other words, there's a slight delay when she makes antibodies. So if we give her RhoGAM before she has a chance to make the antibodies, she'll never make the antibodies. So that way, every kid is like a first kid. So if she never makes the antibodies, we never get over to these two pictures. We always are in this scenario, these two. So with every kid, you just give the mother RhoGAM at birth, and that way she never makes the antibodies, and any subsequent kid benefits and never has to deal with the Rh antibodies that her mom might make. Okay, so that's kind of how you deal with the erythroblastosis fetalis, or hemolytic disease of the newborn. Now, how do you test for compatibility? How do you test blood types? Well, you do it with a card. I mean, in labs you do a little fancier, but this is basically how we do it in a lab situation. You take a person's blood and put 3 drops on each part of the card. And then you're going to mix this drop of blood with, in this one over here, with anti-A serum. So basically A antibodies. Over here, you're going to mix it with B antibodies. And over here, it's anti-D serum, or basically think of it as Rh antibodies. And then you look to see what happens. You mix them up. So I mix a person's blood with anti-A serum, or A antibodies, mix it up. And here, notice I see agglutination. It's clumping up. Well, that must mean then, though, those A antibodies that were in the serum must have bound to some A antigens in the blood and caused the blood to clump. So by seeing clumping mixing with the A antibodies, I know I've got A antigens in this person. If you take that same person's blood and mix it with B antibodies, I can see if they have B antibodies as well, or excuse me, B antigens as well. Well, in this card, you can see that there's no clumping. So the B antibodies must not have had any B antigens to clump with, so we know this person doesn't have B antigens. So I know he's got A antigens, he doesn't have any B antigens, and then when I look at the Rh antigens, notice that the Rh antibody caused clumping. So I know those Rh antibodies bound to those Rh antigens and caused it to clump. And so so this person has positive blood. He's got Rh antigens. So again, wherever you see clumping going on, you know those antibodies you mix the blood with must have clumped with their appropriate antigen, and therefore those antigens are present. So this is A positive blood. And you can do this with all the different types of blood types or on any test. And so I want you to be able to look at the results of a test and be able to determine the blood type. And so I've listed them all here, or this card shows them all listed. And now and basically the result of the test, or what happened in each one of the drops. Now the control is just to make sure that the serums are good. So we don't have to worry about that. They're just showing one. If it did clump, in a control with just doesn't have any blood, we know there's some problem with the serum. So let's not worry about that. So let's take, for example, the top card here. This is one person's blood. So we have 3 drops, ignoring the control, 3 drops. This drop has anti-A serum with that blood. Here's anti-B serum, anti-D or RH. So when I mix this person's blood with A antibodies, there's no clumping. When I mix their blood with B antibodies, there's no clumping. So since there's no clumping with the A antibodies, there must not be A antigens. And since it didn't clump with the B antibodies, there's no B antigens. But the RH antibodies clumped with the blood or caused the blood to clump, which means I know those RH antibodies must have bound up with the RH antigens and caused the blood to clump. So I know there's no A or B antigens, so it's O, but I know there are RH antigens, so the person's positive. And then we can do that for any scenario. So I warn you, don't memorize this, make it make sense. Notice here on this one, I've got clumping in A, anti-A, so I know those A antibodies attacked or clumped the blood, so there must be A antigens because that's the only thing A antibodies can attach to. B antibodies didn't have it, there were no B antigens for them to clump, so I saw no agglutination here. RH antibodies didn't cause clumping, so there must not be any antigens here. So I know I have A antigens, but no B and no D, or RH, which means this person's A negative. Okay, so make it make sense and work through them. Okay, so that's important to be able to do. And that's going to end the video lectures on blood. And so we can talk more about these things. If you don't quite follow how to do these, we can work on through them in class.